Okay, chapter 22. So by that, so this chapter we're going to be talking primarily about the what's called the Keto-Enol tautomerization, and effectively what this chapter is all about is the fact that when you have a acetyl bond O, you have a carbonyl. Okay, what's going to happen is what we're going to do is we're going to take the carbon next to it, as long as it has a hydrogen here. The carbon next to it, we're going to turn that into a nucleophile. So that's basically what we're doing it. And the way that we can do that is tautomerization. Now this isn't really a true resonance structure, okay? Because this hydrogen, right, one of these hydrogens is going to move, okay? And with resonance structures, the atoms can't move around. It's just the pi bonds. But here, you get this equilibrium between the two. But this double bond here, and then later it's going to be a carbanion, will be able to go in and do some nucleophilic attacks with that. So most of the time, it's gonna want to be in this form. Okay, but a very, very small percentage of the time, it's going to be spent. When you have some sort of carbonyl, it's going to be spent in the enol form. Now, the reason why I forgot to mention this, the reason it's enol, is the fact that the ending for an alcohol is ol, right? So and a double bond O, right, is alkene, right? So it's enol, so enol. With that, so with that form. So most of the time, it's going to be formed it's going to stay in the regular ketone form. It's just going to be a very, very, very small percentage of this that's going to be in the enol form. But that's okay. We can work with that. And it turns out all we're really interested in are the carbons, what are called alpha, on the alpha, the hydrogens on the alpha carbon, right? So if you're going to do your Greek letters, so this is from the C double bond O, right? So this is alpha, that's beta, that's gamma, right? So alpha, beta, gamma. So 1 away, 2 away, 3 away. So it's going to be the hydrogens on those alpha carbons there that we're going to be interested in chapter 22. Okay, so the mechanism here, so with it, you can sort of work through it, but basically what you're trying to do is you're trying to get to this enol form, either through the acid or the base. The base will actually pluck the hydrogen away, and that'll collapse down, kicking this up. The hydrogen is a little bit more difficult because you can have negative charges, right? So that goes in attacks, stuff like that. But basically what you're trying to do is you're trying to get into this form here that can then go in and do an attack. So reactivity, it really comes from the fact that another, in this case, resonance form for this is really if you were to go in and have these lone pairs come down, push that out, and so you can have a carbanion here, a stabilized carbanion, which can then go in and do attacks. That's really, that's really what we're driving at with this. So this is a really good really nice carbon nucleophile. Okay, so with that, for the here, it can do everything that a regular nucleophile does. So with that, it's all the SN2 reactions, all the nucleophilic substitutions, and whatnot. Those work great. So with that, so here we go, right? So with that, so we have here plus CH3Cl, right? We're going to have a nucleophile here that's going to come and attack up. So with that, so you're going to end up with CH3 plus Cl minus, right? Same thing here, right? Right. So with that, so here's the alpha, right? So double bond O, right? So with that, so the alpha is right here. Okay, there's no alpha on this side, right? Well, there's an alpha, it's the oxygen. This guy is too far away. All right, so it's just right there. So you have this with chlorine. So with that, so again, this is gonna, in the presence of base, right? So it's either acid or base. Usually you run these in acid, but you can run base there, up there, to make plus CO minus. Okay, so we're having these alpha carbons act like a nucleophile. So there you go. Okay, so like that. So now you can also do this adding so aldehydes and ketones. You can do halogenation right next to at that alpha carbon, stuff like that. So it works similar to the other mechanisms. It doesn't work real well with esters, amides, and carboxylic acids, but aldehydes and ketones are okay. So like that. And so what you end up getting is, let's say you have a ketone here, you mix it with Br2 acetic acid, right? And there's no hydrogens here, right? So like that. So no hydrogens on this side, right? It's all taken up with carbons. You have to have a hydrogen in order to be able to do this here, but there are some here, right? Because it's a CH3 group, so we can react that. So that's going to attack there, and you're going to end up with Br minus there on your alpha hydrogen or on your alpha carbon. So that it worked the exact same way if it's chlorine, works the exact same way if it's iodine. Stuff like that. So here's the mechanism here if you want to do it. Essentially, you're making the double bond, right? So like that, so you're making the enol, which then comes in and does the attack, rearranges to make this. So now, because we can combine these, right? So if we put on a halogen, right? So we right, we can do other reactions with them from previous chapters. So, right, so if we have this, right, so Br2 acetic acid, right, we have it here, we have hydrogens here and here, and we'll talk more about why it would go here in a minute, stuff like that. So it's actually going to prefer to go there, so you're going to go put your Br there. Now if you hit this with base, okay, it's going to come in, grab that hydrogen, that's going to fall down in a nice E2 mechanism. Okay, and so you end up with what's called an alpha beta unsaturated ketone, right? So remember, this is alpha beta gamma. Oop. Like that. So you have a ketone, right, and then a double bond between alpha and beta, so it's alpha beta unsaturated, right, as a double bond, ketone, right, and all in conjugation, right, so 2, 1, 2, right, so with that. So this is really nice. So this is from chapter 7, I think it is, 7, 8, somewhere into there, or you can just think of it as an E2 reaction, with that. So how are we going to do that? We're going to put that here, put the bromine there. Oops, this was Br2, sorry. Put the bromine in, stuff like that. So install the bromine there, hit it with base, boom, alpha beta unsaturated ketone. So it's a nice little reaction. Okay. Now, however, so these work with aldehydes and ketones. They do not work with carboxylic acid, which makes sense, right? Because if you look at what's this reagent, well, that's acetic acid. If this reaction worked, right, your acetic acid would just blow apart all of your, would just suck up all your bromine. Right, so with that, and so what ends up happening is you first have to convert that, right? So this reaction doesn't work. Well, acetic acid, of course it doesn't, because that's what that is. All right, no reaction. Okay, so what you end up having to do is you end up having to hit this with PBr3, and don't forget, this whole purpose in life is to convert that into bromines, right? OHs into bromines. Okay, now you don't have a carboxylic acid anymore. Now you can run the regular reaction, Br2 acetic acid, right? And then that installs the bromine there. When you work this up, this then turns back into your carboxylic acid, because that is so reactive. When you quench it with water, you're going to end up with your carboxylic acid back and your bromine. But you have to go through this PBr3 step first, and then make it into the acid bromide, then you can install the bromine on the alpha position here, and then when you quench it, that's going to be reactive. You're going to get your carboxylic acid back. So with that, so you sort of have to go. You sort of have to cheat a little bit. So with that, make it into an acid. Get rid of the carboxylic acid, do your reaction, and then regenerate your carboxylic acid at the end. Okay, so here's the mechanism, right? All we're doing is we're installing the bromine here. So with that, so you get the you make the enol. So with that, so install the bromine, and then the water will attack. You dump in water at the end. You're just going to do a nice acyl, right? So your water is going to come in, eventually there, up, down, kick away, and end up with your OH. As your bromine's leaving, it's going to snatch a hydrogen away. So with that, so HBr. With that, just normal chapter 21 stuff. So with that, so okay. Okay, so here we go. Right, so we have Br2, so CH. So that's acetic acid, right? So we have an aldehyde here, so we're good to go. So with that, so right, so we're going to install our bromine here. Right? So that's fine. So, but instead we insert that okay. Here again we have a carboxylic acid. Instead we have an OH instead of an H. So in here it's going to be no reaction. Okay. In order for us to do that, right, we have to have Br2 in the presence of PBr3. Then it'll install it, alpha to it. Okay. So you gotta look at what's right there. Now the acidity. So one of the reasons why this works so well is it has to do with the acidity. Okay. So if you look at the pKa chart. Right, so this pKa, it's kind of hard to see on here, but right, so if you look at pKa, carboxylic acid is going to be really acidic, right? Thus the name, right? So it has a pKa of about 5. It turns out that if you have, if you're next to a double bond O, it greatly reduces your pKa, right? So with that, and something like this, where you have this hydrogen, one of these hydrogens here, right? It's next to 2. So with that, so for here, so with that, like pKa is like 9. It's really, really acidic. So with that, so you remember just a regular, CH3H. That pKa is something like, is like, I don't know, it's like 60, right? So with that, so this is. So that hydrogen there is 10 to the 51 fold, so with that, so more acidic. Right, so with that, so it's a 1 with 51 zeros, full, times more acidic. So with that, so with that, so, and even if you just have one of these double bond O's, okay, so something like that, even then, that's that's going to be like a pH of like 19 or a pK of 19, which is pretty acidic, right? So you're talking about that guy there. Even then, that's going to be really acidic. So like that. So it doesn't matter if it's an ester, if it's a ketone, aldehyde. I mean, there's going to be some slight variations, but for the most part, these things are going to be incredibly acidic. So like that. Now, one of the things you have to worry about is you have something. So if you have something like this, if you have two hydrogens and there's a tie, right? So with that, so the first thing you have to look at is say which one's more acidic. So you're looking at here and here, okay? They only have one. They're only next to one double bond O, okay? So that's a tie, okay? So then what you're going to do is you look at more substituted, and you're going to say you want fewer H's on the carbon, That's what you want. So here, this has two hydrogens on it. This side, it has 3, right? So this is the more acidic side. Oops. So this one and this one are going to be more acidic. Okay. Okay. So here you go. Okay, so on this side here, okay, so we just go from one side to the other. Okay, so here, right? So we're looking at here, here, and here, right? Here, no hydrogens. There's no H's, right? It's just all carbon bonds. So it can't be that, right? There's no hydrogens. Look at that. So for here, right, we have it's next to 2 double bond O's. Okay, it's on this side and that side. This side, right, you're only next to 1 double bond O. So that's the first thing you gotta look at. So it's going to be this one here. That carbon's going to have the most acidic hydrogens on it. Okay, because it has 2. It's next to 2 double bond O's instead of just 1. Okay. Now for here, right? So with that, again, we're looking on either side of the double bond, C double bond O. Okay. So well, each of them there's only next to 1. Okay. So here, this one has 1 hydrogen, right? So 1 hydrogen. This one has 2 hydrogens, all right? So because 2 bonds are shown, so 2 hydrogens. Here, 3 bonds are shown, so there's only 1 hydrogen. So that means this is the winner. So that side's the more substituted side. That side's the winner. Be more acidic. Okay. And again, here we're looking at that one. Now on this side, there aren't any alpha carbons, right? Because we need a carbon off of a. We have to look at a neighbor that has a hydrogen on it. I know that there's a hydrogen here, but that doesn't matter. That is on the double bond. We need the neighbor, right? We need it. He says that's not a neighbor. Okay, so that can't be it. So this one wins by default. Okay. So there you go. Now, just to let you know, stuff like that. So, as a quirk of this, stuff like that. So the enol, the neutrally charged species, it's fairly transient. It's fairly unstable. There's no way that you can have a bottle with this. And now you can have other things hanging off of that instead of an H. But if there's just an OH here, you're not going to be able to store it. Now, if there's other things like carbon groups or a phosphate or something like that, that you can do. But for the enol itself, not going to worry about it. Now, if you hit this with base and deprotonated it to the charged species, that's not too bad. You can actually buy this stuff in a bottle. You're going to have some sort of metal, metal counterion here, but that's fine. Stuff like that. But this is more stable than the enol form. Stuff like that. So go figure. So. Okay, so one of the neater reactions here, bring this back, is what's called the haloform reaction. It's kind of neat, stuff like that. So because what can happen is, it's just the normal stuff that you've seen, stuff like that. So, but it works just with methyl ketones. It's a nice test for methyl ketones, actually. So you hit this with base, okay, you're going to deprotonate this one here, stuff like that. So CH2 there. Right, so negative charge. That can come and attack a bromine, right, to give you, to get the substitution there, put a bromine on. One of the quirks is that this actually happens 2 more times, okay, because you're actually activating this a little bit more, right? It's making it a little more substituted, plus it's weakening the bonds because of that bromine on there. And so what you end up getting is all 3 of these original hydrogens getting converted into bromines. Okay. Now it turns out that's actually a pretty decent leaving group, and so what actually happens is because you throw in a whole bunch of sodium hydroxide, this actually ends up doing just an acylation reaction where it goes up, down, and then kick away, and you end up with this. And as normal, right, if there's an H hanging off of there, it'll actually grab it as it's leaving, so you end up with O minus plus H, C, and then Br3. Now the reason why it's so this will work, right? So this works with bromine, works with chlorine, works with iodine. And one of the reasons why it's called a haloform reaction is so if you put 3 chlorines here, that becomes chloroform. Okay, so you would actually make chloroform in this reaction. But this is actually a really nice test, like if you're doing structural determination. This is one of these things you'll hopefully see in organic chemistry lab is a test for this. If you have an unknown, you do this you do the haloform reaction here, stuff like that. So it's a pretty nice little reaction, stuff like that. So the other thing we deal with these is you're going to get you can make these things. These enolates here, they're very nice nucleophiles. They can come in here and do SN2 type attacks. Okay, so if you want to add some carbon groups here, you just have a leaving group on your carbon. This will attack and extend out your chain. Okay, now this follows the same rules as SN2 reactions, right? You want you want the leaving group on a primary carbon, right? This is a fairly large. That's a fairly large. As a nucleophile, you need it to go there, right? So with that, so also, you're going to get inversion of stereochemistry where you add that carbon, right? So with that, so you're going to get inverse stereochemistry. So with that, so it follows all the same rules of regular SN2 because that's all it really is. So with that, so okay. So one of the things that we can do with this is we're going to add these 2 and 3 carbon pieces to extend out our chain. Okay, so with that. So, okay. So, one of the things you can do is you can take what's called this malonic ester synthesis. So, with that. So, this is usually the one that you want to use when it's cheap. Okay. If you hit this with a base such as sodium ethoxide usually. Okay. Right, so these 2 hydrogens are right next to these double bond O's. Right, so with that, so these are going to be really acidic. So that's going to grab that and pluck that away to make this, and so you're going to end up with lone pairs there and a negative charge. Now that can come in. Okay, it's just the reactions we've been doing, most of this chapter, and so with that. Now that's a really nice nucleophile. Okay, and so let's say we want to hit it with some sort of carbon chain or whatever, stuff like that. So let's say we have an ethyl group here, the bromine. This will come in and do just normal SN2 chemistry there, up to there to there, and then you can make like that. Now, if you wanted to add some additional groups here, you could. You just go through this process again. You add more base, that rips this away. You can add an additional carbon group to extend it out there. But here we'll just do the one, but that's totally fine. When you're done with this, if you hit this with water and acid, okay, so acid, what'll actually happen is the molecule itself will decarboxylate, so one of these esters will go away. And not only that, you will also get a reaction here and turn that into an OH. And so what you would end up getting is this. Like that. Now if you can redraw that as 1, 2, 3, 4 as just like this. That. But these 2 carbons here came from that carbon there. Okay? These 2 carbons here came from these 2 carbons here. Okay? So you can track where you're supposed to do that, right? So all we're doing is we're taking this carbon group and extending it out 2 carbons longer with a carboxylic acid at the end. Okay. So this is actually kind of a cool way of making cycles. Okay, you can do it twice. Okay, so again, you're going to have our ethyl. It's just the same reaction. It's just you're hitting the same molecule twice. So you hit this with sodium ethoxide. That comes in, grabs that. Same thing we always do. Double bond there, negative charge. Okay, so crazy thing is now we're going to have bromine, right? Again, just a regular SN2 reaction, right? Coming to here. Right, so we have this 4-carbon piece here. Right, so 1, 2, 3, 4. Right, another bromine. If we hit this again with sodium ethoxide, right? So again, that's going to grab that one. Move that away. So now again, we're going to have lone pairs and a negative charge, but instead of hitting a separate group, we're just going to hit our own group a second time. Hit that, kick that away, and so what you end up getting is basically stitching it together. But it's no different than the chemistry up here. Oops. Like that. So these 4 carbons here, right, are just those 4 carbons there. Now when we hit it with acid, right, what do? Well, we knock out one of those ester groups and we convert that into an OH, right? So it ends up looking like that. Okay. These 4 carbons came from here. Right? These 2 carbons, right, came from here. Just this is no different than what we were doing up here. Okay. It's kind of a cool little way of making a ring. Okay. So for the synthesis, okay, right, so we want to make 2, 4, 6, 1, 2, 3, 4, 5, 6, hexanoic acid, okay. Okay, we need to figure out how to synthesize this. Oops. Okay, so we're always going to have this 2 carbon piece here, okay, and that'll be that 2 carbon piece there. Okay, so that means we're going to need to have, right, so 1, 2, 3, 4. That means we're going to have a 4 carbon piece added to it. Okay, but the steps are always the same. You hit this with sodium ethoxide. Okay, that's going to rip off one of these hydrogens. Okay, now you got to add your 4 carbon piece, right? 1, 2, 3, 4, right? Bromine. Right? So that's gonna attack there. Kick that away. Right? So you're gonna end up with one, 2, 3, 4, right? And when you hit this with H plus and acid, right? Again, you're going to knock that away. That turns into an OH. Okay, there you go. So you have 1, 2, 3, 4, 5, 6. Right? So 1, 2, 3, 4, 5, 6. An acid, right? Those four carbons there came from here. These 2 carbons came from here. Okay, there you go. Now, if you wanted to make an additional branch here, okay, so let's say instead, right, I said, okay, you got to start with this. And instead, you end up having to make that. Again, you have this 2 carbon piece with the carboxylic acid there. That's going to be from here, right? But you're going to have a 4 carbon piece, right? There. And the other thing is, we're also going to need a 1 carbon piece. Okay, so what's going to happen is we're going to have to put on this 4 carbon piece, and then we're going to put on this 1 carbon piece here. Okay, so there's some practical reasons of why you don't want to do one or the other. On a test, I wouldn't care which one you did first. So with that, but again, which carbon piece you put on first. So, but you're going to have to do it twice. Okay, so the first one here, right? So ethyl, sodium methoxide. Right, that's going to rip off. That's going to rip off one of those, like that. Then we're going to hit that with our. Let's do the 4 carbon piece first. 1, 2, 3, 4. Right. So with that, so bromine. Right, so that's going to attack there. Okay. So you're going to end up with. 1, 2, 3, 4. So then you can come in. So okay, so we've got this part put on, right? Now we've got to put on the methyl part. Okay, you're going to go through the exact same process. Again, you're going to hit this with sodium methoxide. Right? That's going to rip off this. Okay, you still have this 4 carbon piece, right? So again, we're gonna react that with, now we've got a 1 carbon piece that we have to add. So 1 carbon with the bromine, that's gonna come in, attack there, to react. So here, flipped right, just hopped right away. 1, 2, 3, 4. Okay. Now, when we hit this with acid and water, okay, again, we're going to knock out one of those esters, and then we're going to convert the other one into an OH, right? So here we go. 1, 2, 3, 4, 5, 6, right? 1, 2, 3, 4, 5, 6. And then off of carbon 2, right? 1, 2, we're going to have a methyl group. So there we go. So the hardest part here is just keeping track of your carbons, and this is where dots will actually help you out, because to keep track of not miscounting your carbons. Okay, so I highly suggest you doing that. With that cell. Okay. Ooh, so another design to synthesis here. Okay. I know we're hitting this a lot, but make sure you guys get lots of practice. So about that. So. Okay. Okay, again, first thing you got to do is figure out where all your parts are coming from. Okay, so again, we're just going to use 2 of those carbons there, and so it ends up being this carboxylic acid and the next one over. So it ends up being here. Okay, so that means our other piece has to be here, and our bromine is going to need to go there, because that's where the connection is going to be made. Okay, but that's so our process, so our steps are, again, it's always going to be the same. Sodium methoxide, right? So that's going to take off one of those hydrogens. Right, so but that way to that. So we have this piece here, right? So those 4 carbons, right? 1, 2, 3, 4. The connection's going to be there, so that's where our bromine needs to be. So that's going to act there. Kick that away. So this is our 4-carbon piece. Now again, once we hit this with acid and water, right? Again, we knock out one of those esters and convert that into an OH. And here we go. Right, so this is where that ester would have been connected through, right? So with that, so there we go. So here's our 2-carbon piece here, and our 4-carbon piece. Okay, so acetoacetate works exactly the same way, except for 2 little quirks. Okay, instead of an O-ethyl group here, okay, we're going to have just a methyl. Okay, the other quirk is that when you get to this step, it's just the ester side that gets on it. So we're going to have an ester and a ketone side and an ester side. It's just the ester side that gets decarboxylated, and stuff like that. So usually it's going to be a methyl group. Stuff like that, but it doesn't have to be, and we'll see some examples here. Okay, but otherwise it works exactly the same way. Okay, you're going to hit this, right? Just imagine that a methyl group: methyl, 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 methyl. Okay, otherwise it works the same way. Okay. Now, okay, so this is a little tricky, but really it's the same thing, okay? Okay, here, right, that's the acetoacetate. It just so happens that these carbons have a couple extra groups on it, right? But again, there's an extra hydrogen right there. There's an acidic hydrogen, right? Because it's between 2 C double bond O's, right? So what we really want to make, right, so with that is just simply that, okay? So this group here, right, is just that group there, okay? Those three carbons there, okay? And then, so we're going to have to add this part. Now, where this connection is between these 2 pieces, that's where the bromine's going to have to be on here, okay? But again, we're just going to hit this with sodium methoxide. That's going to remove that hydrogen to make this, okay? It's no different than what you guys have been doing this chapter, right? So, okay. Then we're going to react that with the connection's going to be between the here and here, so this one needs that bromine on it, right? So with that, so we're going to attack there, kick that away. So we're going to end up with it's no different than what we did up there. Now again, when you hit this with water and acid, okay, now you have a ketone side and you have an ester side. It's the ester side that gets decarboxylated. Now, if this was an ester, it'd be an OH, but it's not. It's just a carbon group, so that stays behind. So there you go. Okay. Okay. Hopefully you can see the rhythm of this thing. Okay. Acetoacetate synthesis. Okay, so again, So 1, 2, 3, 4. It's a 4-carbon piece that we're adding. Okay. So this part here, right, is going to be this part there. Okay. And so what we need is an extra 4-carbon piece there. Okay. But again, we're going to hit this just like all the other ones, right? We're going to hit this with sodium ethoxide. Okay. It's going to rip that away to make this. Oops. To make the carbanion. Okay. Now we have to add a 4-carbon piece, right? The connection between this group and this 4-carbon piece is here, right? So is there, so that's where our bromine needs to be. Okay, so that's going to tack there, kick that away. Right? And you just hit it with acid and water, right? Again, we're going to knock out we're going to decarboxylate it, get rid of the ester here, and then here we are. All right, so here's our 4-carbon piece. Here's the ketone part. There you go. Now, all this is saying is that you can have a huge different types of things. You can make carbanions right next to double bond O's, and some of them are even, even if you have a nitrile group here, that also works. But this can come in and attack various things that you would attack with like an SN2 type reaction. Okay. Now I saw this reaction and I liked it, stuff like this. So. Okay, so like that. So what's happening here? Okay, so you can sort of see this reaction. So if you see this on an exam, okay, so like that. So you say, okay, CH3, CH2, right? Oh, okay. So what's going on here? Right. So it's just an acyl, it's an ester reaction, right? So up, down, kick away. So we're just gonna make that, right? That plus O minus that. Okay. The problem is this molecule is the exact same molecule as this. Okay, so this reaction, I'm sure it does occur, but it doesn't get you anything. Okay, and so what ends up happening is, if you notice, I wrote this 2 here. Okay, so what's actually happening is you have 2 of these molecules. Okay, here, put one in black and one in blue here. Okay, one of these, doesn't matter which one, instead of getting instead of having the ethoxide act like a nucleophile, we're going to have it act like a base. Okay, the ethyl group, just CH, this is an ethyl group, and the ET, that's all that stands for. That's going to react there to make that, to make this. Okay, same thing we've just been doing this entire time. Yep, that's just what's going on here. Okay, now this we're going to use as our nucleophile. Okay, so now this can come in, react. Oh, I'm sorry, that wasn't an OH. That is an O ethyl. Sorry, it's an ester. That we can come in and go up, down, kick away. So what we end up with is here, okay, so we've kicked this away, and then we've made the connection between there and here. So here, like that. So these 2 carbons, right, so here's those 4 carbons, right? So 1, 2, 3, 4. There's just been a nucleophile to come in here and do this, right? Because remember this reaction, right? A nucleophile coming in, attack, up, down, kick that away. It just so happens happens to be one of, this is the nucleophile. So this is the nucleophile, comes in, attacks, up, down, kicks this away. So now this part here is connected there. So here's your product that's formed. Kind of crazy. Stuff like that. But it's kind of a neat reaction. So, okay. Okay, now biological alkylations. Biology loves this reaction. Okay, stuff like that. So now it has. So what will happen is you'll have this. You'll have a base. So something like this would bind inside of get inside of a binding pocket, and there would be some sort of base, which is typically either aspartate or glutamate, or excuse me, yeah, it's not glutamate. So for that, so yeah, it's going to say glutamate or aspartate. So with that, so that would come in there, and you can rip off that hydrogen, right, to leave to leave the enolate form here. That can go in and attack, do whatever attack. So if you have S-adenosylmethionine, the adenosylmethionine part, that's a good leaving group. But what you can do is you can take it'll detect that methyl, and the homocysteine, S-adenosylcysteine homocysteine group here acts as a leaving group. Okay, it's just a biological bromine, right? So like that. So it's just a leaving group that goes away, and you're left with a methyl group there. So with that, so they do this all the time. It's a great way of making carbon-carbon bonds. Stuff like that for biology. Stuff like that. So, stuff like that. So, okay. Good luck.